Good evening, students. Good evening, students. Good evening, doctors. I hope you are preparing well for your NEET quiz exam or national exit test, whatever it may be. And many of the students are preparing for the future NEET quiz exam. All are welcome here. You can share this video with your friends so that large number of people will be benefited with these videos. So today we are going to discuss the microbiology subject. This microbiology subject is a very important subject because about 20 to 25 questions come from here, and about 5 to 10 questions of the infectious disease part in the medicine, and 2 to 3 questions in pediatrics come from microbiology. And PSM questions, most of the PSM questions can be easily answered with the knowledge of microbiology. So this subject is very important. If you master this subject, you will be scoring very good marks in your NEET PG exam and the national exit exam. So with this, let us begin with the first question. Draughtsman colony are seen in with A. Anthrax, B. Pneumococci, C. Pertussis, D. Yersinia. Now, draftsman colony are seen in pneumococci. They are carom coin-shaped colonies. They are seen with pneumococci. So, due to alpha hemolysis, colonies of pneumococci resemble colonies of Streptococcus viridans, but on further incubation, the colonies of pneumococci become flat. They become flat with raised edges. They become flat with raised edges and central ambonation. Flat with raised edges and central ambonation, so that the concentric rings are seen on the surface viewed from the above. This is known as carom coin or Draughtsman appearance. So the correct option here is B. Pneumococci. Granulomatous, granulomatosis infantiseptica is caused by A. Pseudomonas, B. Chlamydia trachomatis, C. Group D streptococci, D. Listeria. Granulomatosis infantiseptica is an intrauterine infection of newborn. Granulomatosis infantiseptica is an intrauterine infection of newborn which is caused by Listeria. The predominant feature of this condition is extensive focal necrosis, affecting especially the liver and less often the lungs. The mortality rate is very high. Occasionally, meningitis may be associated with it. The organism can be isolated from the affected areas in child and frequently from genital tract of mother, who may or may not manifest the disease. So, granulomatosis infantiseptica is caused by D. Listeria. Now, stalactite growth in ghee broth is due to the following. Stalactite growth is due to the following. It is Yersinia pestis. So the correct option here is A. So the cultural characteristic of Yersinia pestis, please listen carefully. When grown in a flask of broth with oil or ghee on the top, also called as ghee broth, a characteristic growth occurs which hangs down into the broth from the surface, resembling a stalactite. Whereas on nutrient agar, the colonies are small, delicate, transparent disc, becoming opaque on continuous incubation. On blood agar, colonies are dark brown due to absorption of hemin pigment. On McConkey agar, colorless colonies are formed. In broth, a flocculent growth occurs at the bottom along the sides of the tube. So the correct option here is stalactite growth in ghee broth is due to Yersinia pestis. Milk ring test is done to detect milk ring test is done to detect which organism present in milk. A. Bordetella, B. Brucellosis, C. Bartonella, D. Salmonella. The correct answer here is B. Brucellosis. For detection of brucella in infected animals, pooled milk samples may be tested for bacilli by culture and for antibodies by several techniques. In the milk ring test, a sample of whole milk is mixed well with a drop of stained brucella antigen and incubated in a water bath at 70 degree for 40 to 50 minutes. Please listen carefully. In the milk ring test, a sample of whole milk is mixed well with a drop of stained brucella antigen and incubated in water bath at 70 degrees for 40 to 50 minutes. If antibodies are present in milk, the bacilli are agglutinated and rise with the cream to form a blue ring at the top, leaving the milk unstained. If antibodies are absent, no color ring is formed and the milk remains uniformly blue. So Bordetella is detected using this. So the correct answer here is brucellosis. Superantigen cause superantigen cause superantigen cause A. Polyclonal activation of T cells, B. Stimulation of B cells, C. Enhancement of phagocytosis, D. Activation of complement. Superantigen, this question is very, very important. This has been repeatedly asked. Superantigen causes polyclonal activation of T cells. Superantigens are capable of activating up to 20% of peripheral T cell pool, whereas conventional antigen activate 1 in 10,000. So it is A, polyclonal activation of T cell. Respiratory syncytial virus causes all: A. Coryza in kids, B. ARDS, C. Bronchitis, D. Common cold. Coryza respiratory syncytial virus causes all: A. Coryza in kids, B. ARDS, C. Bronchitis, D. Common cold. The correct answer here is B. ARDS. RSV infection leads to a wide spectrum of respiratory illness. In infants, it can cause pneumonia, bronchiolitis, and tracheobronchitis. In this age group, illness most frequently with more with rhinorrhea, low-grade fever, often accompanied by cough and wheezing. In adults, most common symptoms are common cold with rhinorrhea, sore throat, cough. It causes severe pneumonia in elderly. Sinusitis, otitis media, and worsening of chronic obstructive reactive airway disease are associated with RSV infection. Microorganisms which cause pyogenic osteomyelitis. Microorganisms which cause pyogenic osteomyelitis. It is B. Staphylococcus aureus. Now the organisms causing pyogenic osteomyelitis. Staphylococcus aureus causes 80 to 90% cases of acute pyogenic osteomyelitis. Staph aureus has a receptor for collagen which contributes to its pathogenicity. Other organisms causing osteomyelitis include Escherichia coli and Pseudomonas in intravenous drug users and patients with urinary tract infection. Haemophilus influenza and group B streptococcus in neonates. Salmonella in patients with sickle cell disease. In sickle cell disease, Salmonella causes osteomyelitis. So the correct option here is B. Staph aureus. In nutrient agar, concentration of agar is A 1%, B 1.5%, C 3%, D 4%. So the correct option here is B 1.5%. Nutrient agar is a general purpose nutrient medium used for cultivation of microbes supporting the growth of wide range of non-fastidious organisms. Nutrient agar is popular because it can grow a variety of bacteria and fungi and contains many nutrients needed for bacterial growth. Nutrient agar is made by adding 1.5% agar to nutrient both 0.5% peptone, 0.3% beef extract, yeast extract, 0.5% NaCl, distilled water. pH is adjusted to neutral 7.4 at 25 degrees centigrade. So the correct option here is B 1.5%. Which cells cause rosette formation with sheep RBCs? A. T cells, B. Tuberculosis, sarcoidosis, C. D. ulcers. It is T cells, which is found in Daigeor's syndrome. A. Tetany, B. Eczema, total absence of T cells. It is A. Tetany. Now, tetany. Complement formed in liver are complement formed in liver are A. C2C4, B. C3C6, C9, C3C6, C9, C. C5, C8, D. C1. Complement formed in liver. The correct action is here C3, C6, C9. C3, C6, C9. Complement formed in liver is C3, C6, C9. Biosynthesis of complement components. Intestine C1, macrophage C2, C4, spleen C5, C8. C2, C4 intestine. C1 C1 is in intestine, macrophage is C2, C4, spleen C5, C8, and C3, C6, C9 in the liver. Hereditary angioneuritic edema is due to hereditary, hereditary angioneuritic edema is due to A. Deficiency of C1 inhibitor, B. Deficiency of NADPH oxidase, C. Deficiency of MPO, D. Deficiency of propyridine. It is deficiency of C1 inhibitor. Hereditary angioneuritic edema is due to C1 inhibitor or C1 esterase inhibitor deficiency. So correct answer is A. Opsonization takes place through it is A C3A, B C3B, C C5A, D C5B. It is C3B. So the correct option here is B. Staph aureus causes A. Erythasma, B. Chancroid, C. Acne vulgaris, D. Bullous impetigo. So the correct option here is Staph aureus causes Bullous impetigo. Bullous impetigo. Impetigo is divided into two types: non-bullous impetigo caused by Staph aureus and bullous impetigo caused by Staph aureus. Non-bullous impetigo is caused by Staph aureus and Streptococcus pyogenes. Bullous impetigo is caused by Staph aureus. Which group of streptococcus grow at 60 degree? Which group of streptococcus grow at 60 degree? A, A, B, B, C, C, D, D. It is group D streptococcus, which is heat resistance and grows at 60 degree. Fish tank granuloma is caused by M. marinum. M. marinum. Fish tank granuloma is caused by M. marinum. Correct answer here is M. marinum. Fish tank granuloma, also called swimming pool granuloma. Fish tank granuloma, also called swimming pool granuloma, is caused by M. marinum. Aquarium granuloma, also known as fish tank granuloma, swimming pool granuloma, is a rare skin condition caused by non-tubercular mycobacterium known as Mycobacterium marinum. Skin infections with M. marinum in humans are relatively uncommon and are usually acquired from contaminated contact with contaminated swimming pool, aquarium, or infected fish. Aquarium granuloma presents as slow-growing, inflamed, red bump, nodule or plaque at the site of infection. It is characterized by painful, itchy skin lesion, some ulcerated or crushed. Skin changes usually appear roughly 3 weeks after aquatic exposure. If the condition is acquired from an aquarium, then the dominant hand is often affected. If this condition is acquired from swimming pool, then symptoms can also develop on trauma-prone body zones such as elbows, knees, feet, and tip of nose. Infection can be painful or painless and may be life-threatening. They can be classified into 4 types. Type 1, lesions in form of crusted or ulcerated nodule. Type 2, distributed inflammatory nodule spreading pattern. Type 3, deep infection. Type 4, lung infection and other systemic manifestation. There are usually long delay from symptom manifestation. Aquarium granuloma is relatively non-specific in clinical position. Therefore, more common skin conditions like cellulitis, foreign body reaction, skin cancer, and fungal or parasitic infection are often explored first. Overall diagnosis and treatment of this rare skin infection is often delayed because of lack of suspicion. Lab tests for finding M. marinum include cultures, where a swab or sample is taken and grown in laboratory. Cultures of M. marinum are fairly difficult to grow, and results may take several weeks. The culture may be negative even if there is an active infection. Treatment may still be considered even if test results are negative, especially if the patient history supports past fish or fish tank exposure. In the absence of positive culture result, a skin or tissue biopsy may be helpful to render a microscopic diagnosis. Another technique that can be used to detect bacteria is PCR. A biopsy use itself is sufficient to detect the exact type of bacteria by means of PCR. How to prevent this disease? Avoid fresh or salt water activities if there are open cuts, scrapes, or sores on the skin, especially in water where this bacterium is known to exist. Wear heavy gloves, leather or heavy cotton while cleaning or processing fish, especially fish with sharp spines and that may cause cuts to the hands and skin. Wash hands thoroughly with soap and water after fish processing or use a waterless cleanser. Wear waterproof gloves while cleaning home aquariums or fish tank. Wash hands and forearms thoroughly with soap and running water after cleaning the tank, even if clothes were worn. Ensure regular and adequate chlorination of swimming pools to kill the bacteria that may be present. Treatment: treated with monotherapy of antibiotics, clarithromycin, trimethoprim, and ciprofloxacin, with deep infection being treated with combination of antibiotics like ethambutol, rifampicin, doxycycline, and minocycline. Antibiotic treatment time varies from 1 to 25 months with a median treatment of 3.5 months. Botonase fever by A. Rickettsia japonica, B. Rickettsia conori, C. Rickettsia siberica, D. Rickettsia australis. Botonase fever is caused by it is Rickettsia conori. Rickettsia conori. Rickettsia conori, also called Mediterranean spotted fever or Botonase fever. Botonase fever. It is Rickettsia conori. Which one of the following is called praesenocard bacillus? It is C. Pseudotuberculosis. Praise-Knockout bacillus. C. Pseudotuberculosis. Darting motility which occurs in Vibrio cholerae is also found in Shigella, Campylobacter jejuni, Pneumococcus, Bacillus anthrax. Correct option here is B. Campylobacter jejuni. XDR-TB is defined as resistance to fluoroquinolones plus INH plus rifampicin plus amikacin. So the correct option here is Hebranos is caused by Frisch bacillus. Frisch bacillus. Now what is Hebranos? What is Hebranos? Duodenal aspirate is used in diagnosis of Giardia lamblia. Izumi fever is caused by Yersinia pseudotuberculosis. Unsegmented eggs are seen in which parasite? Trichuris tuchura. Brazilian purpuric fever is caused by Haemophilus aegypticus. So with this, we conclude with the microbiology MCQs. These are some of the important MCQs which are likely or most likely to come in the examination. So please be thorough, watch the video again and again. You can WhatsApp me directly on the number which is flashing on, and you can click the link in the description box to join the Neet PG Masterclass, which will be beneficial for you in future. I wish you all the best. Take care.